I'm Andreas, also from Stanford. I'm a lab member of both the Andreas and Steinmetz labs, and today I'm going to present a basic research vignette on how to use IGVF catalog components to understand non-coding variant function. Specifically, how to use enhancer gene predictions that we've now available in the IGVF catalog to link GWAS variants to their target genes, as well as to identify predicted cell types of action. So as you know, you're all aware, there are thousands and thousands of GWAS variants that have been linked to various phenotypic traits, including disease risks. And most of these variants are in fact located in the non-coding part of the genome. And so studies have, for instance, found that autoimmune disease GWAS variants are predominantly located in enhancer-like elements in CD4 positive T cells, showing that variants, we can expect them to impact gene expression programs through perturbing cis-regulatory elements such as enhancers. And, of course, knowing that a variant is an enhancer, that's already a good piece of information. However, that brings other challenges that we need to overcome to really understand what that non-coding variant is doing. And these challenges include, we need to know which gene is regulated by the enhancer that this variant perturbs, and as well, also importantly, in which cell type that regulatory interaction takes place in order to know where and how a given non-coding variant affects gene expression and downstream cellular programs. And so our solution or our proposed approach here, in order to provide a resource to the community that allows researchers to quickly query different non-coding variants and find out what might be good hypotheses for target genes and cell types of actions, is to provide a resource of genome-wide maps of cell type-specific enhancers and their target genes. And so in this here example, so this shows sort of like what I mean by that. So assuming we have a risk variant that has been linked to a disease like a GWAS study, for instance, and is somewhere located in the non-coding genome here, somewhere close by to a gene which might or might not be the gene that variant regulates. And then we have, for the different cell types we're looking at, we have different annotations. So we have these tracks that show in the little boxes annotated enhancer elements in the genome of these cells, including some molecular assays that have been used to characterize these elements, such as, for instance, DNA accessibility measured by ATAC-seq, which is often used. And most importantly, we have the connections that connect these enhancers to their target genes. And as you can see, in that case, what that would allow us to do is to say, oh yeah, that risk variant here, that is in an enhancer in cell type 1 that is regulating this gene. Therefore, that cell type and that gene are good candidates to follow up on to know more about what that non-coding variant might be doing. So the question, of course, is now, how do we actually get to these maps across many cell types and throughout the genomes of these cells? So CRISPR perturbation screens have become very powerful tools lately to perform full perturbations of regulatory elements and then read out the effects on the target genes, allowing us to systematically map these connections. However, they still lack the scalability to do this genome-wide, and especially across many different cell types and tissues. Some of them might just simply be inaccessible for experimental manipulation. However, the data collected by such screens allow very new and exciting novel ways to use them for predictive modeling. And so this brings us back to the IGVF map perturb predict framework that has been introduced earlier. And so we're just going to quickly show you how we can use that to build enhancer gene models, or short EDG models. And for the mapping component, what we're doing here is we collect a bunch of different genomic assays and features that we know are informative for enhancers and connections to their target genes. We're collecting them across many cellular models and tissues. We then also collect genomic perturbations datasets from the aforementioned CRISPR perturbation screens, but also eQTL and MPRA datasets, and then bring those two together in order to build new predictive models where we can use perturbation screens as ground truth datasets to train models that then can predict, based on the functional genomics data, what are enhancers in a given cell types and which genes are they most likely to regulate. Another big component of that modeling approach there is also we can use the genetic perturbation data to systematically benchmark the performance of these models, which we believe is a very important part, especially because we want to know what is the expected accuracy of these maps these models are then going to produce. And so this is an ongoing effort right now in IGVF. And we have our hands on several models, and then some of these models that we anticipate to be in the catalog soon, or predictions from these models to be in the catalog soon, are the single-cell E2G and GODAR E2G models. So these are both classifier models that have been trained on CRISPR perturbation data. So meaning for these models, we used enhancer perturbation screens as ground truth data to teach those models how to predict enhancers and target genes from other molecular features. And then there's also PGBoost, which uses a similar architecture, but is trained on fine map EQTL data as a ground truth training center. And as already mentioned, these models is a really big part of our efforts. And so we have different benchmarks that we can run with the different genetic perturbation data sets. And here's one example that shows benchmarking E2G models against CRISPR enhancer perturbation data from K562 cells. So here we're specifically asking the question, how can these different models, or how well do they at identifying enhancer and the target genes that have been experimentally inferred via these CRISPR interference perturbations? And you see this here on this slide, a comparison of different models we looked at for both models that can operate on single cell input data. So that's, for instance, model that make predictions based on single cell multiome or ataxic, as well as on the right bulk models that take bulk data such as DNA sequencing. In each of these plots, you see the precision and recall performance metrics shown. So simply speaking, that means the higher curve is, the better a model performs at predicting the CRISPR enhancer perturbations or enhancers to the target genes. And it's good to see for both of these cases, we see that the models that we use here in this effort, single cell E2G and GODAR E2G, perform really well and perform better than a simple distance to TSS baseline predictor, as well as other models we've compared them to. And so overall, this makes us very optimistic that we can build high quality enhancer gene maps, and especially those allows us to gauge the accuracy of the resulting maps that we can build with these models. So now that we have these models in our hands, so how do we use them to actually build these regulatory maps? So here, that forward is relatively straightforward. So essentially, we start out by computing the features. So what that means, we across all the cell types we want to make predictions for, we compute the model features such as DNA accessibility at candidate elements, but also things like expected 3D contact of elements or candidate enhancers with candidate target genes. We then put these into the trained models that we've trained previously using the perturbation data. And then they, based on those features, predict cell type specific enhancers and the predicted target genes. And these predictions then allow us to build these maps of enhancers and target genes across all different cell types, which then, as previously shown, allows us to intersect those with variants, to annotate non-coding variants, and prioritize enhancers that they perturb, as well as target genes in specific cell types. So to summarize, the effort of building E2G maps in the IGVF catalog has the goal to build an integrated and searchable resource to understand non-coding variant function by identifying enhancers, target genes, and cell types of actions across a diverse set of cell types and tissues. And so now for the second part of the talk, I'm going to walk you through some of the examples and show you some of the first versions of these that we have currently available for the public on the IGVF catalog. And so currently we have roughly 92 million enhancer gene predictions across roughly 1,500 biosamples spanning more than 350 unique cell types and tissues. And to say more is to come, we'll have soon more models as well as more cell type states and tissues. You can access predictions for these enhancer gene predictions on the various parts of the IGVF catalog, namely the variant pages, gene pages, region pages. So these are pages that show a summary of what's available in the catalog for specific variants, genes, or genomic regions. And as well, we have an E2G app that allows to easily visualize these predictions in a genome browser. So for our little exercise here, let's consider a use case that's a realistic way we think people might interact with the catalog. So let's assume we have a non-coding variant of interest that has been linked to a trait by a GWAS study. And so we essentially want to figure out, what are the predicted target genes of that variant and in which cell type might that variant regulate said genes. So in our example, we're going to use this variant RS875741, which has been linked to mean corpuscular hemoglobin via GWAS. And that trait is essentially how much hemoglobin red blood cells contain. And so we're now going to use the IGVF catalog to figure out what target genes that variant might regulate. And so this is terribly easy by basically you just copy the RSID, put it in the search box of the landing page, the IGVF catalog, and you hit go, which takes you to the variant page. So this is the page that has all the information that this variant has been annotated with in IGVF. And if you scroll down a bit, you get to this table of enhancer gene predictions. And so this table contains now one prediction per line. And each line is essentially one case where the variant intersected an enhancer that was predicted to regulate a given gene in one of the cell types or tissues that are part of the IGVF resource. And so that table, contains a couple of columns, which most of them I think are very easy to understand. So of course, it's a cell type, so that's where the regulatory interaction is predicted to take place. The target gene, so the gene that this variant is predicted to regulate. The score from the E2G model. So typically a higher score means more confidence. The enhancer coordinates, so these are the coordinates of the element the variant is perturbing. A link to the data set. So in this case here for the ENCODE E2G model, that will take you to the ENCODE portal, allowing you to look at the exact input data that was used to make the predictions, information on the model used, as well as the type of element that this model uses or models, as well as a distance of the variant to the gene, which is useful for any sort of biological interpretation. We also allow to download that table easily as a, for instance, a .csv file, which you can easily use to more advanced queries and downstream analysis. And so by looking at this table, we basically see that this variant here that is linked to MCH regulates a bunch of different genes, but something that already immediately pops out that a lot of these interactions are predicted to happen in the hematopoietic multipotent progenitor cells, which of course makes sense because these are the cells that later on give rise to lineages leading to for instance, red blood cell differentiation. One of these genes that shows up is CPEV4, which a quick search shows that this gene has been shown to be linked to erythroid differentiation, but also lipid accumulation in the adipocytes. However, here with our predictions, by looking at this table, we can see that this variant is specifically linked or predicted to regulate that gene in the hematopoietic progenitor cells, which is a good candidate for where this variant might affect MCH. We can then easily subset the table by just putting here in the search box hematopoietic multipotent progenitor cells to see all the predictions in that specific cell types, and that shows there's a bunch of genes that variant might regulate in that cell type. We see that there are also NSG2 popping up, which is a gene that has been in some way associated as well with hematopoiesis. However, for our example here, we're going to stick with our first nomination of CPEV4. And so if you want to know more about that gene, we can just simply click on the gene name, because that is a link which takes you to the gene page, which is a very easy way to look at the cis-regulatory landscape of a given gene of interest. And so we can now see here in this E2G table there, we can see enhancer gene links for that genes across all the different IGVF cell types. And so when we subset that, for instance, again, to our cell type of interest, we can, for instance, look at a more in-depth view of the predicted regulatory landscape of that gene in our cell type of interest, which might be used to, for instance, identify additional regulatory elements and interactions that might regulate our trait here, MCH. And so I think, this shows a quick overview of how we can use these different tables and pages in the IGVF catalog to relatively quickly get an idea of what a given non-coding variant might do and which cell types might be interesting for downstream analysis and maybe experiments. As mentioned already previously, we also have the E2G app though, and this app is particularly useful to visualize enhancer gene links in the genome browser, and it's easy accessible as part of the IGVF catalog apps. So if you go back to the landing page and scroll down, you will see that the app, alongside other apps such as the coding variant related apps that have been presented yesterday, and then you just simply click on the tile E2G, which takes you to the E2G app, or you can also access it via this link here directly. And when you go to that app, you're again greeted by a search box where you can look up your variant, and which brings up a table that is very similar to what we've seen before, with the addition that now here there's this little plus sign that allows you to add predictions to an IGV session. So we're going to select here again. Here's our prediction for CPEB4 in the hemopoietic progenitor cell from before, but now we see, like in this case here, there's also a prediction from the single cell E2G model in bone marrow proerythroblast cells, which also makes a lot of sense since those are a later stage along the differentiation into red blood cells. So we add those to the IGV session by clicking on these plus signs, which then populates this nice genome browser view where we can see at the green line the position of the variant, annotated enhancers in those cell types, as well links to target genes, as well and accessibility from AttackSeq. And so this very nicely shows how we can use the app to quickly look at these things in a very intuitive way, and also scroll through the genome to get an idea of regulatory landscapes around variants, genes, or regions of interest. So now I've shown you how we can use specifically the enhancer gene links in the IGVF catalog to better understand non-coding variants and prioritize predicted target genes and cell type of actions that might be worth looking into for downstream analysis or experiments. However, there's of course much more to understanding non-coding variant function. And so as part of building the IGVF catalog, we are working towards adding a lot of different products in addition to that, and some of them are already available in the catalog. And so here's a very brief overview of these other anticipated catalog data sets and analysis products that will be useful to study non-coding variant function. So starting from the bottom of the whole causal cascade from variant to phenotype, we will have variant effect predictions of how variants affect TF binding at regulatory elements, both chromatin QTL and expression QTL data sets that allow us to gauge how variants impact chromatin landscape and activity at elements, as well as gene expression programs. Single cell multiome or single cell pack data that allows us to look at, again, the impact of variants or diversity variability in accessibility across cell types, and then effects on expression. Genomic perturbation screens from MPRA, STAR-seq, CRISPR interference, and variant editing screens, allowing to look directly at direct experimental evidence, how sequence changes, variants, and elements impact chromatin activity, as well as expression of target genes, and also perturb-seq studies that allow us to trace how non-coding variants that affect a given target gene can have transacting effects further down the regulatory networks and impact cell types and cell states along the way to impacting the phenotype. So to conclude and to summarize what we've seen in this presentation, I've shown you that we can use genome-wide maps of enhancer gene regulatory interactions to quickly query non-coding variant function and make hypotheses about which genes might be regulated by a variant and in which cell type that regulatory interaction might take place. The IGVF E2G resource in the catalog currently contains predictions for roughly 1,500 biosamples that span more than 350 different cell types and tissues. You can access these predictions easily via the variant, gene, and region pages, as well as through the E2G catalog app that allows easily visualization of EQG links in a genome browser style. And we will have more models and cell types and states and tissues coming very soon. So keep an eye out on that, as well as there are more anticipated additional non-coding data sets and analysis products that will come soon to the catalog, allowing you more additional ways of how to query non-coding variant function and use it for your work. So with that, we would like to invite you to go ahead, go to the catalog and start, looking up your favorite non-coding variants. And, hopefully that's useful for your work. And please share any feedback and then feature requests that you have with us, because we're really eager to improve the resource and make it so that you can get best use out of it for your work. So with that, I would like to thank everybody involved. So everybody in IGVF, it's a tremendous collaborative effort, of course, NIH for funding, including our program officers who constantly give feedback and then help us navigate this, as well as many different individual contributors from affiliated labs. And looking forward to addressing any questions and taking feedback after the end of the presentation.